to go. Attila, it's good to see you.
It's very nice to see you. It's great to be here. Thanks for the invitation, Christian.
Thanks for taking your time. I know that you have a lot on the plate yourself. What I find interesting when I went through your material, you are one of the rare people who not only are in academia, but also start a company. So you work in the intersection of academia and entrepreneurship.
Yes, indeed. So I think it's getting, so some of my friends are doing that. And then the question is, I think in many cases, is that how much they balance between the two. And that's, I have seen various versions on how some colleagues who also started a startup company, some moved full time there, some kind of more like an advisor for the company, some really a CEO, and then they can spend time here, spend time there. I'm trying to balance between the two. So I still have at the university a little research group who is kind of working on a bit distinct questions than what we work at the company. So I'm trying to manage both sides, which is challenging indeed.
To frame it for the audience a little bit deeper, you're then working not only in academia and entrepreneurship, your focus is on what I find particularly interesting, digital twins,
Yes.
artificial intelligence, and how computational biology is changing the entire way drug discovery works.
Indeed, that's the direction we started our startup company. And then just to have a sentence on what we do in an academia. So I think what we do there that's was called computational systems biology and still called computational systems biology. And now we do experiments in the lab as well. So that's more on the systems biology side. And there kind of we also do digital twins, but there we do digital twins of yeast cells and yeast colonies. So that's really basic science. And then from some of these ideas, we started to use on human simulations first, and then combine those with AI technologies to get to a level there where we could build first a digital twin cell, kind of that how a cell functions, and if you perturb with a compound, how it will respond. And then from this digital twin cell, we create now more, we can simulate cells from 29 different tissue types, and that kind of builds our digital twin patient. And this digital twin patient, then we can see that if the drug or the drug candidate or compound of interest can reach these different tissues, then how the molecules, the molecule complexes, the protein complexes, how they come together, or how the regulatory pathways are perturbed by a given compound. And we combine this with AI to understand that how these changes in different tissue levels, how this might reflect in side effects and actually effects on people.
A lot to talk about today, but before we dive deep into these topics, I know that many people on the internet today are in a constant hurry. And could you please frame for those who are impatient and need a reason to stay, what they miss if they move on now?
Okay, I think we will discuss about how we can use such digital twin approaches, kind of combining simulations and AI technologies to better understand how a compound can cause side effects in humans. So that's the major goal which our company is working on, really focusing on side effects. And then we are also moving this to the next levels when we simulate a population of such digital twins. And then I think I will explain more on the technology, that how this works and how this could be used for the pharma companies. And probably there are some people in the audience who are working in the pharma companies and I hope that they are looking for solutions like ours. I really hope they will hang around and listen to what we discuss. And I hope they will get the message that if they approach us, then we can show them an angle, and we can connect kind of really early stage drug discovery when you even look for only the targets, or you have the drug candidate compounds, the multiple of them, or 50, hundreds of compounds of interest. Then already at that stage, even if you haven't synthesized these, we can already do some in silico approaches to predict that if eventually this compound might reach humans, then what type of side effects this might cause. And this is kind of that where we try to connect the really early stage of drug discovery to really late stage. And that's, I think, what we will be mostly talking about.
That's fantastic. How can I imagine that? What problem do you solve in the pharma industry?
Well, basically the problem is that there are multiple levels of this problem. First is to identify that if there is a disease, that which could be the target, to identify which target protein might need to be inhibited, or which protein target needs to be modified by the compound. And then if you have the target, then you have to identify, okay, which might be a good molecule that will be able to do the modification of interest. And there are a bunch of companies who are working on this target identification and compound lead optimization, and how these molecules should work to have the right effect, what they're looking for. Our technology, in principle, can allow some of these. So for target identification, some aspects of target identification we can also address. We can also check that an on-target side effects, which target might have the least on-target side effects. So we can help in target selection as well. But the majority of right now what we could do is more in the next step. When you have multiple compounds which you want to test, then we can help them to save money and time to test all of those on either for cell lines, eventually on various animal models, and eventually before it goes to clinical trials. So this is the whole step which we would like to overarch, and there are a bunch of tools that can be used to predict that how it will affect liver, how it will affect kidney. There are some of these specific modeling tools as well, or AI tools for specific side effects or more kind of adverse effects. But to have a broader view of what all possible side effects could be there, that's, I think, our unique selling point, if you wish, where we can explain that what could happen. And the extra piece is that since we do this through a simulation, so it's not full AI and as a black box, but there is in the middle, we have a simulation and a molecular mechanism. So we can also track back that if we identify a side effect, we can tell that this side effect might be caused by changes in given protein complexes or pathways. And we can also track back that these changes might be connected to one of the off-target proteins. So when you design these these drug candidates, then the main goal at the beginning usually is that, yeah, it has to reach the protein of interest. But this might reach other proteins, either with the same warhead, so with the same part which you design to reach the given target. But it could be that some other side chains or other parts of the compound might reach other proteins and might interfere with that. These are what's called off targets, and that's where we can track back that how a side effect might be caused by a combination of some protein complex changes and pathway changes, and how these might reflect back that which off target might be causing that side effect. So this could help eventually the pharma company to decide that if there is a side effect which they might want to avoid, then they can go back to the chemoinformatician to redesign the molecule or select from the pool of molecules one which won't have the given off target which could cause that.
That's interesting. What I always have a problem with imagining is one thing. So when I hear to you speak, when I listen to you speaking, and when I hear you speaking, I can imagine it with molecules that are already on the market, and someone wants to use it for new targets. And we also work with well-known targets. You have a lot of data, you can match that and have more accuracy. But for me, in drug development, the holy grail of drug development is novel target, novel molecules, novel biologicals, yes, now I have it. And to match that, novelty, something completely new. killing a new disease with a novel drug nobody else is doing yet. Huge market. Everybody dreams of that, especially when we talk about investing. What I have a problem with is it's the same with quantum computing. When you solve that problem and say, okay, look, you have this new target and you have this new compound substance and you simulate it, how can people be sure and confident that the results are right? I mean, it's entirely new.
Yes, yes. This is always kind of the problem of how much data you can feed into your
Mm,
models. And in our models, what we feed in, and we keep feeding and growing the data, what we're using for our training. So one of the first big challenge is to identify that which might be the target and the off targets of the compound. So which proteins it might bind to. So that is kind of a large question, which is kind of approached by a bunch of different companies, and we are also developed a version for that. And our goal is especially that we are looking at more than 8,000 proteins. So
mm,
we test against a bunch of 8,000 proteins if a compound binds to either of them. So these are the proteins where there is something about their structure is known. Either it's known or it can be predicted by one of the tools that can be used for that. So I can imagine that this problem might be solved in a few years by some players much larger than us.
mm.
Point to isomorphic labs, for instance. So I can imagine that they would come up with a solution that how this can be solved in an easy way. We have a solution which I think, as I said, that our goal is to really identify all possible side effects. So that's why we are trying to get all possible off target. So we might optimize a bit to identify, maybe I hope not overshoot, but to identify all possible side effects in all possible off targets and eventually all possible side effects. So there the challenge is that how much you know about the structures of the proteins and how much you know about the structure of the given compound and how they match to each other. But that is something where there is a lot of development. Well, there were Nobel Prizes and such. So that is something which I feel that will be solved soonish. Kind of the middle part, and I think that's really our unique point is this cell simulator or digital twin cell, where we simulate that. So the basic idea is that what we're simulating, that how proteins come together and form protein complexes, and how these, and if we know the abundance of proteins in cells, we can simulate different tissue types with different protein abundances. And if a protein, so most of the proteins have multiple potential binding partners. So if you have protein A, which can bind to protein B and C, but protein B is present in much higher abundance, then you might see much more of AB complexes than AC complexes. But eventually, if there is a compound which you design that will inhibit B, then eventually you might see the AC complexes to appear. So that's kind of the underlying technology we look at. And in that sense, kind of how much you can trust in this, to go back to your question, is that how good is your data on protein-protein interactions, which is again a bit related to the earlier question, that how much you know about the protein structures and their interactions. So in a way, this PPI network, this protein-protein interaction network, there are multiple sources where data so there are a bunch of databases where information on known protein interactions are there from various yeast to hybrid tag, so various experimental data on that. And there are also a bunch of predictions. So the AlphaFold mold former bolts could be used in that direction. So there are multiple tools which go into that direction. So that is growing this day by day. In a sense, what we used, as I said before, that my lab is working on yeast. So I won't say I'm a yeast expert, but I'm probably I used to be a yeast expert, and now I'm somewhere between yeast and human. And what is known about the yeast, so why it's good, but this was the first eukaryote that have been sequenced, and there is much more known about it, not by quantity, but by percentage. So the protein-protein interaction network of the yeast is much smaller than the human, but much more complete our knowledge. So many of these simulation tools which we have, we test or train it on the yeast data, where we know much more on them. So we are now actually working on a manuscript kind of to show that how well it works in yeast. And kind of we train how to set up the whole approach on the yeast data and use it on the human data. And in that sense, I would say as more data is coming, it's getting more and more trustable. I think, and that's why what comes in at the last stage, that when we're looking at the side effects, so we can identify a few hundred side effects where we believe that our predictions can be strong. There are some where we do not have enough information. So that's why I would say that this will, as we'll have more data, it's getting more and more trustable, but it will be more and more trustable. Multiple different questions can be addressed by that.
And your team doesn't want to stay solely on the academic side and answering academic questions and say theoretically it would work. You also want to use it in the industry. What bottleneck in drug discovery has your team found?
Yes, so one of the bottleneck is, especially on this, so to explain the next step of our pipeline, that once we have these simulations, we identify that in which tissue type, which protein complexes and pathways are perturbed. So this we were running through on more than 1,000 drugs where we know what type of side effects they can cause and which have been out in the market. And then we train a machine learning algorithm to learn that if this protein complex goes up in liver, that pathway is less active in intestine, then it's diarrhea. So in this type of way, we kind of learn that which molecular perturbations can be connected to which side effects in which tissue type. And what is a limitation there is really that we can train this on clinical trials where we see the results, which is most often the successful ones. So unsuccessful clinical trials are rarely released. Now in Europe, they start to push into this direction. So that's why clinical trials are done elsewhere first. but that could be one thing which could help a lot and could enrich that. Now we are not just, so now we can predict well those side effects which can appear at multiple drugs, which are kind of easier accepted side effects on drugs which are released to the market. Of course, side effects can be, there is a different level of acceptable side effects for a drug which you take for headache and which you take for cancer. So a cancer drug with a little bit of diarrhea is usually good enough if that cures cancer. If that is causing this, you have the same side effect for a headache pill, that you might not want to have that. So in this way, we still can learn some of the more serious side effects, which can be, diarrhea was not the right example, but especially some heart and brain related side effects are the ones where what we started to see in these lines, where neurological and psychological side effects, what we see to be able to predict, because these are appearing in drugs which are out on the market. We also collected less than 100 drugs which have been withdrawn. So there we know at least the reason they have been withdrawn. And then we are, in our training, we take this into account and we ensure that that's something which we are kind of worth scaling. And then we are sure that those should be able to predict this whole system. But really the desired thing would be kind of to get from all pharma companies, all results of all clinical trials and train on that. Because our simulations might also contain some of those biological features, those changes in protein complexes and pathways, which might cause abrupt heart attack, which would stop even in mice, and then it would never reach clinical trial. We might still see in our simulations, but in our training set, we do not have enough data to tell that, well, this symptom can be connected to that. So actually we are kind of also considering to go into this direction to identify reoccurring changes in the cells, which we cannot connect yet to side effects, but kind of identified which might be the side effect, which was not in our training set, but still could be a relevant one.
Why is this so important for your team to get data from failed clinical trials?
Well, because pharma companies, they want to understand that what will be the result of their clinical trial. They try to rule out everything to have it as a failed clinical trial. But if you could tell them in advance that from these 10 or 15 compounds, because before you have the DC nomination, which of the ones which might go into the direction of failed clinical trials, which might go to the going through, that would open an even stronger business line. So I think that now we are confidently can tell that from those side effects, which eventually can be seen in some of the drugs, which of those you might gonna see from your compound. But we might be able to tell that some which cause really a failure of the whole drug development, but that is, since there is not enough data, that is more questionable in that sense. So that is something where I think our technology is there, we just need the data.
What must happen that we get the data?
Sorry?
What must happen on the market that data becomes available?
Yeah, well, basically pharma companies need to change their mindset and figure out that if we share data with the others, that eventually could help all of us, which is not really the trend so far, let's put it that way. So pharma companies really want to keep all their earlier failed drugs, which they probably never use, but they steal all data they don't release. So if they would just release those which eventually they stopped full and they kind of given up to use any of the derivatives of that, because that would be already a problem for that to release, that would already increase this training set for us.
What's the benefit for pharma when they release the data and they give it to you? What benefit? How could you describe the benefit to someone, decision maker, executive in big pharma who has not much time to listen to you? How would you describe it in 2 minutes? What's the benefit giving for him or for her? What's the benefit for them giving you the data?
Well, now I have data on successful clinical trials, so I can predict that how your successful clinical trial, what will be the results on those side effects which can be seen. If you give me the data on the unsuccessful ones, I would be able to predict better than if it will be unsuccessful or not.
Okay, so without data, prediction is impossible.
I won't say impossible. That's why I put better, because now we can still predict a bunch of the side effects, which are, it depending really on the indication. So for some indications, you can see that if there is some heart-related disease appearing there or some strange neurological one, that could kind of stop the development of a compound. So if we see multiple of those, that could. So in a sense, I would say that our tool right now, I believe this will improve a lot in the coming years. We have already some ideas how, but right now, what is the most useful is if you have really these 5, 10, 15 or whatever number of compounds to compare them. And then we can tell, well, from this pool, these are the ones where we see kind of mild things, not neurological, not heart, not related to bad things that can cause your failure. Whereas these ones might show some signs that might not lead to good things. So I think that's the current stage of what we can do, is to, on the selection, to tell that which looks better. We won't tell that, well, this will fail because of that.
What's the big vision that you have, assuming that you get all the data you want?
Yes. So the one thing where we're going with this is that right now we are running this on an average patient clinic digital twin. The next thing, actually, we are opening a funding round to create a version of this, which is more like a population, where we would create a large number, tens of thousands of digital patients, where we consider the genomic distribution in a population, so all the SNPs and their combinations. So we generate an artificial population where we have multiple of the SNPs which are associated with some side effects. And then we add the compound to each of these digital patients to identify that which genetic marker might be connected to a given side effect. So that could really help now at a later stage, more in the clinical trial, in patient stratification, that, well, even what we have currently, that could help that what might happen at clinical trial, but at that stage we can tell what might happen at clinical trial with whom. Mm-hmm. So that's kind of one of the direction we go. And as we have more data, what will happen is that we will have more and more type of side effects which we will be able to predict with high confidence. So now we have it, this is around the level of 500 or 700 of side effects from a list of, well, actually it's more those which are more often appearing, that's in the number of 1,000 or 2,000. The whole list is 10,000, but there are some which probably never happens. But so we can predict a large portion, but this will increase.
Did I understand it right? Your core goal with your technology is focusing solely on the side effects of potential new drugs.
Yes. So actually, we can go further eventually. So I would say that if this digital twin works, then right now the perturbation which we give is more by a compound that perturbs our digital twin cells. But we can also do, and actually we submitted an EU proposal more in this direction, where we'd use this digital twin now to have a perturbation by molecular level changes. So transcriptomics or proteomics data on a disease or on a set of patients. And then what we could do is to use the same core machinery of predicting that how protein complexes and pathways are perturbed by these changes in protein levels. And then we connect it not with side effects, but with diseases. And then to identify that this given patient with these molecular profiles, what type of side effects this might, what type of disease might be seen from this data. And of course, this can be combined with other approaches, with other features can be added to this whole training set, like, blood pressure and blood measurements, and whatever is collected, metabolomics and so on, whatever is collected on a given patient. So this can be getting more, it could be one of the portions of identifying that what could happen with a given patient, or at this stage might be healthy, but we can predict that what type of patient this person might be.
So the side effects are just the starting point into the future.
Yes, so I would say that the side effect is one of that's kind of the major goal of what we do. And then there are these, what we now put as more kind of research proposals, where we kind of try that what else we can use this technology for. So the main line of the company is really for side effects, and then we have these side projects which are more on okay, how else ways we can use this. And eventually staying on the side effect side. So I mentioned that we would create this population of 10,000 digital patients with different genomic compositions. So eventually from there, and that's really kind of the future vision, where we can go is we can, so if we can create 10,000 patients with different SNP distribution, then we can look for a single individual with that person's given SNP distribution. So if we have the genomic profile of a single individual, then we can test that if we add drug candidates or a given drug to this given patient, how this patient will respond, what type of side effect this patient might show. And this could go now in a different direction. So that's more not drug development, that is more towards the clinics. So it would be more as a decision support system then we will need all the regulatory approval for software as a medical device. When it gives a support for the medical doctor, the physicians, to decide that for this given patient, I can give these various compounds or the combination of them. We already tested that, so it's really easy to add multiple drugs. So in this case, it would be more kind of drugs which are on the market, but then which combination or which individual drugs might be the best for this given patient that could be simulated basically with the same technology. But then it would run most probably on the clinics on a server, even connected with probably with a tool that can give a proteomics profile or genomics profile, ideally both for us, because then it would work even better for the given patient. And then we'd split out that, okay, what is the best treatment for this patient, or probably kind of that some ranking, and the physicians decided which one of these to eventually use.
That's great. So when I look at the value chain in drug development, drug discovery,
Yeah.
where in the value chain do you position yourself currently at this point in time?
Okay, at this point in time, we are really at the early stage, and that kind of we connect the early stage with late stage. That's the jump which we're trying to do. So one is in the exploratory phase, we can help kind of the target identification and tell that which target might have the least on-target side effect. So that's kind of one of the possible applications. The other one could be really at early stage discovery, when multiple compounds would be tested on cell lines or animal models. And before doing that, to select that which you might want to throw out or which you want to select based on this profile, what we give, that which might be safer. And even in clinical trials, as we discussed with some of the earlier pilot partners, that they say even in pilot, so if we do it in before clinical trials, well, you can also have the DC nomination, so before clinical trials, but also during clinical trial, it can give, so if you have a single compound, that's what we heard, that it's still good to know that what they should prepare for. So even if they won't stop the development of the compound, which is quite late stage, but if they have in advance knowledge that, okay, well, we should expect that some people might have diarrhea, but probably no one will have headache. So then that could help kind of the design of the clinical trial. So that could be another direction where we could help.
So the goal at the end of the day, at your current stage, is to help pharma companies or biotech companies from the number of, or the set of molecules they have, to eliminate those which are likely to cause a lot of side effects, and focus their resources and attention on those molecules that show the least possible side effects in your simulations.
Indeed, that's the direction, and that's kind of can be done at different stages, because there are multiple steps when you have such selections for larger, smaller number of molecules, and then kind of to decide which might be the lead, or even to redesign the compound based on kind of feedback coming from us.
Why is this so important?
Well, because the pharma companies lose lots of money kind of by running experiments on compounds in early stage, which eventually they might throw out, because when they push a lot of money into it, it goes to clinical trial and it shows some serious bad side effects, or too many side effects, or those ones which at given drug development cannot be accepted. And then they lost a large amount of money, and they have to go back and select another one from the earlier stage and do it again. So that's time and money.
Did you ever try? I mean, I know it's hard, and I'm always really surprised when I discuss on the internet or in direct messages how few people understand drug discovery and drug development at the end of the day. So I would like to spend a little bit more time here to also help the general population understand what it really takes to move something from the lab to the market. And it's not easy. So your goal at the end of the day is to reduce the likelihood of failure, time and cost connected to that. And my question to you is, I know it's hard, But did you ever try, or did your team ever try to calculate in a number, in a cash number, and in months or in years, what your solution could help to save at the end of the day?
Yes, so yes, and we gave up, and then we started to discuss with a few companies who can do that.
Mm.
So actually, we have a price quote for such calculation. But we have on the advisory board a few people. Actually, one of the earlier CEOs of the second largest Hungarian pharma company, and he is advising us on this. And now we are looking into one company who said that they would be able to do this type of calculation. So now we are negotiating with that.
Yeah, but because basically when I think about the numbers that I heard in the last 20 years, it ranges from $1 to $5 billion per drug, and time to market is somewhere between 5 to 15 years
Yes.
at the end of the day. And the likelihood of success, Christoph Länger was on my podcast in 2021, and he repeats this number over and over again. He says, when you look at really early stages, really lead candidate selection, he said, one of 10,000 makes it to the market. So 10,000 molecules fail, one makes it to the market.
Yeah, so we need to identify those 9,999.
This would
Yeah,
be the, I
so
mean, this would be a great thing.
yeah, so that's really the issue, and now there are a bunch of approaches, especially on AI, to design the compounds in such way that generative AI to avoid this and to be successful, which kind of starts to work nicely. I would say there is more and more use of AI and related computational technologies in to make the compound efficient and to do what they really want to do. I have seen less of the approaches to go towards side effect avoidance, because in a way, kind of that comes too late in the whole process, and well, biotech companies can sell what they have already at earlier stage, and well, there are of course there are a bunch of even computational approaches, the pharmacokinetics, pharmacodynamics, and a bunch of calculations that how this compound might behave, and quasar can tell toxicology. So there are a bunch of approaches to kind of rule out the really basic issues that could come, which eventually might be in vitro studies, they would show up. So that is something which you can avoid by running some of these other approaches, which a bunch of companies offer. There are some who are really doing this really well, and even now FDA can accept some of these in silico approaches instead of real experiments to identify that how the compound will metabolize or something like that. So there are approaches on these lines, but to really have kind of the final list of what are the possible side effects, that is something which I haven't seen from others yet.
Mm. When we look at an idealized world, I mean, one part is the development from lab to approval for a clinical trial. And the other part is then the clinical trial section from phase 1 to the market at the end of the day. And your approach helps to narrow down the number of candidates, or to improve the number of candidates that hit the late stages of preclinical trials. So this is your current market position.
Yes, and what we've seen as a challenging part, that if we talk to larger pharma companies, then we talk to the toxicologist whose problem is really clinical trials. Then they love our approach, and they said, "Yeah, this is cool. I would like to be interested in this." But then eventually they figure out that, well, we have to convince our chemists, because they are the ones, the medical chemists, who are dealing with this at this early stage selection. And then they connect with them, us with them, and then they are, we are optimizing for efficacy, and that's our major goal. And in this way, that's what we see as the medical chemists. Of course, they kind of consider side effects, but then we come with our our magic tool. They might also do some bioinformatics and some with our some chemoinformatician. In fact, with many of these pharma companies now, we talk with the pharma chemoinformaticians who kind of understand our approach, and they come up with the most relevant questions in that sense. But and then eventually, that's what we've seen, that we start to deal with these two type of people, and eventually what we need, and that's kind of the critical part, to reach the person above the whole thing, who looks at the whole pipeline and who can tell that, well, to avoid this late failure, now we have to use this approach in much earlier stage.
This is the direction, exactly the direction in which I want to go. These different mindsets, which I find so interesting in drug development, because it's so hard to bridge it. And you nailed it. In clinical development, you need a safe and effective drug that can also be tailored in a way, in a product that patients adhere to the treatment and it fits into the healthcare system. When I go earlier into basic research, but basically chemists say it's cool, we have a new molecule, and that's it. I mean, this is what they need to do at the end of the day to get the publications out, to gain, it's simplified, of course, but to gain credibility. And in preclinical development, basically, you start thinking about the first time, okay, is it effective? And you want to work on maximizing the efficacy of a potential drug. The worst side effect that I can imagine is basically death. I mean, it's a side effect. At the end of the day, you have
It's
an effective.
on the side effect list.
Yeah, but you have an effective drug, but at the end of the day, it kills the patient, so it doesn't really work on the market. My question to you is, how do you deal with this with people when you see, okay, they have their goals to develop new molecules, and side effects are not really their problem, but it's really important for the person above them to have this ruled out. How do you motivate chemists to use your technology when it solves none of their obvious problems?
Yeah, so in principle, what we've seen that we are dealing with smaller and larger biotech companies, and that's, I would say, that's where it works much easier,
Mhm.
because there they work on a few compounds, they want to select the best one, and there their interest is to be able to tell that what will happen if eventually this might goes to clinical trial, because either they develop it further or they want to sell it to a pharma company, and then if it fails with the pharma, that's not good for them. So that's why they want to get this, and it's much easier to reach the CEO and talk to them directly. Whereas as we go to larger and larger pharma companies, it's much harder to reach that given decision maker who can see the whole pipelines. So that's why it's kind of, we are trying to kind of get to salespeople, advisors, and such, who opens the door to get to talk to those people. And in some cases, really now what we tried, and in some cases it started to work, is to talk to those toxicologists or some other people who have the pain point, what we can address, to bring it inside to the level where they can really address this.
Yeah, it's really challenging. I totally understand that to
Yes.
move it forward.
Yeah, so that's really to so that's why we've seen that with smaller biotech companies, it's going okay-ish. We have a bunch discussed with a bunch of them, and they really like the approach. We get lots of feedback. We have dealt with a few of the mid-size pharma companies, and there we could reach basically those chemoinformaticians, and there we get to a level where they understood, and they said that, okay, this looks nice. They gave us kind of some examples which were kind of, so what's the other challenge from our point of view, that what pharma companies are willing to share as a pilot is some compounds which failed for them, because that's less risky to show to some crazy Hungarian bioinformaticians. And when they give it to us, then we give some predictions that what type of side effects they might see, what is the biology behind, and so on. And then they found that, well, we found some of the crucial ones, and then in some cases we did not find, but they said that, well, we stopped this in animal model because in mice it showed something. We said it did not show in us. In human, we see something else. So that's why we were also considering to kind of, we need to create a mouse version, because sometimes pharma companies, they want to see what happens in mice because that's what we have seen, but we are predicting not what happens in mice, but what happens in human. So actually, we had some discussion with the Jackson laboratory because they have a lot of data on mice, and then how we could kind of connect the two things. So we had. So I hope that after summer's gone, then we can go back and kind of draw down the details of this project, which we are considering, that how we kind of translate from mice data to human data, kind of in our framework.
I mean, the animal models are basically a bridge to the human model at the end of the day in preclinical
Yes.
trial. And when you talk about digital twins, to expand a little bit on this topic, you are talking primarily about creating human data, human twins.
Yes, and as I said, that we have these yeast twins kind of to test the tool and kind of to have a quick test case. And we also have kind of one or two couple of cell lines, and we have, I think, one data source for mouse. But that is something where we might gonna go depending on how these discussions with Jackson Lab or others might go on, because that's what we see, that we said, well, eventually everyone is interested about human, but they can see the proof in mice because that's cheaper for them to test. So so we might need to do the mouse, not because that's really interesting for us or for everyone. It's just needed to do because that's kind of how we can prove for them that, okay, we can catch that as well. But many times what you see in mouse is not the same as in human. Well, that's kind of one of the problems there. But that's why we might need to get into the mouse, especially to prove for them that yes, it works. Even in those cases where you can see. In one thing which I see that could be really useful in that sense is really that in mice we might see the same biological mechanisms. And since our tool is especially good in identifying mechanisms, that's kind of the idea how we can move between mouse and humans, that the same molecular changes might have different physiological response in mice and in human, and then that's what we could translate in this way.
Okay, now I understand it. I think this leads back to the initial point that we made about skepticism on the market, that you can predict, can you really predict the results? And you said then that when you use a mouse model, it's easy and a cheap study they can do. You can run it in parallel, and you can validate it with your technology, or they can validate your technology and say, look, I mean, give us the setup of your next study in mice. We simulate it in our tool, and then you can run it in reality and then compare the output and see what happened and what did we predict.
Yes, and in principle, this could be also done in cell lines, and then it could be human cell lines or organoids. So in other approaches, and well, there is always this problem that what type of so as we say that now we train in humans on side effects. So what is the training set in mice? Well, there we have something like side effects, so kind of physiological response for various compounds. On the cell lines, it's or organoids, it could be more on the molecular level, but that's why our tool is nice, because we capture the molecular level. So there, if we create a digital twin organoid, we would create that, then kind of the output is really that how the proteins and complexes and signaling pathways are perturbed, and then we won't be able to tell what is the side effect. Well, kind of we can run this same thing on our digital twin human, and then we can tell what is the side effect, but we can kind of show that, yes, our approach can match to your experiments. And in fact, kind of right now, what we show as a proof of how this works is more on these lines, that when we identify a side effect, and then we show that this side effect can be connected to which molecular details in our simulations, then we kind of look into the literature and identify, yes, indeed, this makes sense. So we kind of do the validation post the simulations. We show that, yes, these are fully make sense from a biological perspective. So kind of we do the validations on the, again, back on the protein level. So that's because it's much harder to do a validation on clinical trials, then you have to run the clinical trials. But on the molecular level, you can see that if there is any evidence that this protein complex might be connected to that type of disease or failure on whichever organ.
Yeah, we should not forget that this technology is really in the early stages. So I think 20 years ago, it was not even possible to think about it.
Yes. Yeah, so it's really kind of depends on the data and on the computing power, because kind of to run these simulations. So how the whole company started. So my background in systems biology, I had some of these ideas. That's how these complexes might form and how we can predict the protein complexes. And we created a really clumsy simulator which was running 3 days on a single GPU. And then when I returned to Hungary, I met István Reguli, who is now the CTO of our company, and his expertise in high performance computing. And then we started to discuss that how could we speed up this in a reasonable fashion. And because now we have tens of millions of molecules forming hundreds of millions of reactions, and this is something which we want to calculate in such speed that right now our whole pipeline, if you get a compound in a week, we kind of report the final thing. But now we're going towards to make it as a software as a service, kind of to run it straight. So that's why it's speed up is really a question, and that's kind of where we have kind of part of the uniqueness is coming from that part, that we have the technology and the people who can develop this real fast.
I find it really interesting. You said that you started in systems biology and there you worked the first time with GPUs. Can we make a walkthrough of your career? Because I think it's really interesting to understand how this technology evolved from the early stages to where we are now, and then we can expand on the future. In which year did you start to work with GPUs?
Well, yeah. So the GPU so as I said, I'm kind of by training, I'm a bioengineer. So the GPUs, it was really coming from my student who worked on, and he was running it on GPUs. I understood more or less the structure and what it's doing, but I was not the one who could put it on the GPU. So my background is really I would say more in these intersections of biology and computation. So I was working in places where I was considered a biologist, and in other places I was considered a computational guy. And I'm kind of neither. So, but this first GPU version, to answer really your question, that was kind of 2014, 15, where we had the first version of the simulator. And that time it was really, we were simulating how protein complexes come together. There was no kind of that drugs can come in. And that's why that was really the question which we discussed with István, where it was the speed up, that, well, if this would run in a reasonable time, then we could be able to test how a compound might interfere with this. Because we need to run, so it's a kind of stochastic simulation, so we need to run 100 simulations to have a distribution without the drug and with the drug, and compare the distributions to ensure that how much changes are there. So it's really requires a lot of computing.
Why was this so hard in 2014, 2015 to work on GPUs? What was the bottleneck back then?
Well, it's probably that we were not that much experts in that. So at that time I was working in a Microsoft Research Center, but it wasn't really the Microsoft Research for computation and systems biology. So it was not really the place where we had really expertise on GPUs. So it was a bit learning by doing type of thing. And that's why I came back to Hungary in 2016. So in 18 or so, 17, 18, that's when we started to have this faster version, which was kind of developed by the student who was kind of co-advised by István and me, who is now the VP of engineering and who runs the whole development of the company right now.
Where was the Microsoft lab?
That was in Italy, in Trento.
In Italy,
It's
yes.
a beautiful area.
Why was Microsoft so interested in this type of technology back then?
Yeah, so there are still a bunch of Microsoft research centers all around the world, and I think at that point computational systems biology started. Well, the systems biology term was originally released by von Bertalanffy 100 years ago, but in 2001 Hiroki Kitano wrote a paper in Science, I think, where it was that he was coming up with the concept that how systems biology might work. And at that time, there were a couple of other papers where from leading scientists that, okay, what is systems biology? And I think that was kind of big boom of how that works. And there were kind of aspects on the experimental side and the aspects on the computational side. And of course, if there was something where computation is happening and it's really hot field, then Microsoft started to get interested. And they opened this institute in 2006, and I started there in 2007. So I was one of the early PIs in that institute. Mm-hmm. So it was really Microsoft Research, also in the one in Cambridge, which was the European headquarters where I was a couple of times. So there were a couple of projects which were kind of used as a playground. Now I think they're more focused, but at that point it was really that, well, you do something for Microsoft in-house in Cambridge, but there are some, you can play, and if you really like it, then it might become something from Microsoft. And this research center was really a joint cooperation between the local University of Trento and Microsoft Research. And it was more kind of it was a non-profit organization, and it was really focusing on creating new computational tools and ideas and I think Microsoft's role was kind of to fund it, and I think they had the rights of best offer. So if we create something, then they could buy that. But that was kind of the major connection to that. And I think the institute still exists, but I think now it's not fully associated with Microsoft. I think they might be still connected in some level, but I think in the name, I think there is no Microsoft anymore.
There's a nice side story. I find it particularly interesting that the big companies in the United States like Microsoft, Alphabet, Apple, Nvidia invest also in areas that are not typically their core business. And when something comes out, as you said, they just pick it up and develop it to the market. And if not, then it was a nice try and we move forward.
Yes, in fact, kind of one of my earlier postdocs from Trento worked with Amazon and now with Google. And with Google, he's at this the Moonshot company where they do some kind of similar type of approach to come up with something for bioinformatics, but we do not know yet.
Isn't that interesting? I started working in drug development in 2006, and back then, I think, at least in my environment, nobody thought about Alphabet, Amazon, Microsoft in terms of drug development.
Yeah.
And 10 years forward, 2014, 15, 16, they started investing. And when you look now at the landscape in 2025, I think they start dominating the space.
Indeed. So that's why I say that Microsoft had some early signs in this. They had a biocomputation unit also in Cambridge as well. So that's where was our best connection. So I had a bunch of interactions with people there as well.
And then in 2018, you said you moved back to Hungary, and then your approach changed. What improved in Hungary?
Yeah, so actually, just to make things more complicated, I also had a job in King's College London, in the Randall division there. So in Microsoft Research, I was the biologist. In the Randall Institute in King's College London, I was the computational guy, because that was more a biology research institute. So I had a small group there, and for a while I was working between Italy and the UK, and then kind of my Italian leg I put back to Hungary, and then I was working back and forth between the UK and Hungary as well, and had kind of two labs, but there were more interactions in those directions. And then in Hungary, what was nice is really this university where I am, this Pázmány Péter Catholic University. So where we have this Faculty of Information Technology and Bionics. And here we have a bioinformatics MSc program. And in principle, kind of before coming home, I was asking some of the friends around, okay, which is the nice place where good students are. And they said that this is a good place. And indeed, there are really great students who kind of both capture the computational part and also the biology they know. And in fact, now our company, there are 16 people, and I think 13 had at one stage in their life they were affiliated with this university. Well, it also helps us kind of to kind of collect the talent and kind of get the best master students to do first internship and then eventually start to work with us. So that's kind of a good connection in that sense, and I hope that and I believe that this also works for the university as well.
Yeah, that's definitely an upside when you have a startup close to a university. You can basically select the best from the students and then directly offer them a job. This is a huge advantage.
Indeed, that's kind of works nice. And now we started to hire also people from other places. So now kind of we kind of reached I hope that stage that now they're looking for us even from outside. But the early setup was really coming from this place and alumni and people who we knew from this loop.
When in your journey, so it started in Italy, Microsoft Lab, you moved to Hungary, you worked in between, in parallel, UK and Hungary. Uh, when did you start thinking about putting your idea into a startup, into a company, into a corporate shell?
Yeah, so actually when I was in the UK, then we had a startup company in 2014, I think, which lived until 2018.
Mhm.
And but that was really coming out from as really a side project. So that was the three of us. We made it with two colleagues from Cambridge, because we were organizing a meeting in the Royal Society, and we created a matchmaking algorithm to match scientists to have kind of speed dating, but with an influence one, where we kind of put together those people who might have related knowledge, but they never co-published papers, more or less. That was the simplified algorithm. And then we had this meeting there, and everyone loved it, and they loved this approach. And then we were asked from the scientific journal eLife to write about this in news. We wrote about it, and then the Times Higher Education, the Harvard Business Review were writing that, yeah, this is how future will go. So then we kind of filed in a patent, we established the company, and then we had some deals with the local university, we had with the European Molecular Biology Organization, we organized a couple of meetings for them. But this was really not biology what we were doing. And eventually we did not have an investor, so we were bootstrapping that. And we had some kind of income, but that was not enough to really hold us up. And there were interest to use this, but there were not enough paying customers who would really pay a decent amount, which could really hold a team of a couple of people. So we had one person hired, and we couldn't hardly pay that single person. So eventually we shut down the company, and we learned a lot from it. So you have to be sure that there is a paying customer who is willing to pay for it. Actually, it seems there were, because I've seen recently a company getting a huge amount of investment into this. So it was not that crazy idea, but probably because we were all three scientists, so there were no one with financial or business background. So I think we are doing it a bunch of things in a bit of a clumsy way, and but it was a nice learning curve, because now all three of us are CEOs of various startups. One of them just got 7 million investment in UK. The other one is now looking for a couple of millions in the US. So I think we learned a lot, all the three of us, from this experience. And now I think we all do something much closer to home, and something which is really the biology related and on our major research lines.
So you basically founded Tinder for scientists.
Yes,
Yes.
in a way, yes, that was the Tinder for scientists, indeed, and I think it really worked, and it could have worked if we would have enough passion to it. So I
Mm.
think that was that. Well, this was not really the major line what we're interested. So I think now in our current companies, all three of us, which have much more passion, because this is something which we would do anyway.
Let's double down on these 2 points, because I think it's really important for young scientists who wants to found a company. And currently, it's I think everybody wants to have a company suddenly. Uh, Two learnings. You said one is financial and business background. How would you phrase that for a young person who says, I want to found a company? Why is that important?
Yeah, well, if you have time, then do an MBA, but and time and money, but that's not the fastest and easiest way. Uh, Actually, you can learn a lot from TED Talks and just by asking, not ChatGPT, so one of the GPTs, or 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 read a couple of books. So there are very nice books on these lines, which eventually can give you. So in a way, eventually you will have someone in your team who will be much more experienced than you in these business lines. You should have you should have the vocabulary to understand what they're talking about. When first I had this, that okay, what is a difference between a business plan and a financial plan, that kind of stuff, that which I also had to learn. So to get clear on a couple of things, and to have good mentors. So now there are a bunch of accelerators which provide a bunch great mentors. And this is, I think, what could be really useful, that if you get to one of these accelerator programs, you get the mentors who kind of tell you that, okay, read this book, look at that video, and so on. And then that could help really speed up the whole thing to get into this direction. And now they really prepared for that. The other approach could be, and that's I've seen in Hungary now, they started this type of incubators when they try to match basically a scientist from the STEM field with someone from economics. And then they say, okay, well, if you can talk to each other, then maybe you two will be the core of a startup.
When we look at, when we stay a little bit with finance and business, at the end of the day, I mean, you said it, hiring is a great idea. A CEO should build a team and not try to do everything alone, but he or she needs a basic understanding. And when I look at the universities, I think business skills, especially in the STEM field, are still underrated. I don't see many curriculums that say, okay, one outcome of our students might be they found a company, so they should at least have some basic know-how. How can we improve that? What's your opinion?
Yeah, so I've seen on a bunch of universities. So on in Cambridge and MIT, they have these innovation programs where kind of students come together. They get, in fact, our project in Cambridge was in there, where students get a project from one of the faculty members, and they kind of help the faculty members to work out the business plan and the whole business idea for that. And actually now our university, Pazmine, we had a joint degree with Notre Dame University and on an entrepreneurship, and now we also have kind of a separate course for innovation management and innovation. So that is something which I see at multiple places. They start to do something. In some places, I see that there are some governmental help into this, that they create a program, a mentor network that can help these students. But and in our university there was one project which is now a startup, and this started really as in the innovation course, so it could work. Well, the faculty who are teaching there, who are mentoring, have to put a lot into this. But it is worth it, because you see these bright eyes of the young people and their enthusiasm, and that's just makes your heart warm.
No, absolutely. It should be a basic skill, even in schools, on earlier. And I was just smiling because you said there are some government programs. I grew up in the 80s, and I'm always getting a little bit skeptical when governments try to teach entrepreneurship.
Yeah. Yeah, so yeah, well, the good government program and the government funds to have the mentors,
Mm,
and
mm.
then mentors do the job. So
Yeah, yeah, that's great. The second point that you mentioned is passion. That's an important factor when founding a company. Why, in your opinion, is this so important?
well, because it's full of failures, and you still need to stand up. And then if you do not have passion, then you give up at the first point. So that's one thing. You have to believe in what you're doing, and you have to believe in it, even if you go through a bunch of failures and no one else believes it. Then you still have to push that. Well, so you have to test yourself, that do I believe in it as much as if everyone says that I'm the stupidest person in earth, I would still believe in it. And if you do that, then you can say that, well, it's worth to do it. I will have the passion to push this through until the point when others starts to believe in it.
Do you think passion and love are synonymous? terms.
Passion and
Passion and love. Love.
in this aspect could be well, I think it's I would put passion in it's love plus enthusiasm in that in the
Mhm.
sense that you love it, but you are willing to do a lot to it, which you could do also for love, but it's you have to actively do it. That I think is a bit more than only I love this project. I have to be enthusiastic about this project.
Mhm. That's very well put.
Willing to give up things to do that.
That's true. That's true. Good explanation. My question to you is, you said that for your first company, you were not as passionate as your current company. Um, how do you decide what's going on inside you that you make the decision consciously and say, I'm passionate about this, and it's not just a fling. This is the reason why I asked the question about love. I mean, people fall in love all the time with other people, and then after 6 months they find out not a big laugh. It was just some chemicals playing funny games inside me. What's your decision point where you say this is really my passion area? I love it. I'm enthusiastic. I get going, and I will keep going, even if people say I'm stupid.
Yes. So, yeah, you have to believe in it. You have to believe in yourself and your team. And you have to build a team in which you can trust and which you say, well, we will be able to do this. So that's kind of part of the passion. And it has to be something which you would, so what you have to think that 90%, more than 90% of startups fail. So you have to do this in a way that even if it fails, it's worth it. So if you do it for, yeah, I will be rich and that's why I'm doing this, then you're not passionate. So you have to be passionate to do it that, well, something nice will come out even if it fails. If you are willing to do that, then it's fine. If you're just willing to do that, well, eventually I will sell it and I will be a millionaire, then yeah, then you will fail earlier. Because there will be multiple failures when investors reject and such, and then you will start to think, okay, do I really need to push? No one believes me. What's next? So in a way, that's kind of the way. I would say where love comes into this. Love is more on the investor side, I think.
Mhm.
So you have to be so passionate that you make investors fall in love with your project. And if they are in love with your project, then they will be willing to finance it.
How do you make them fall in love? What's your secret?
Yeah, well, if I would know, yeah. So then I would be.
I was hoping that you have the secret, the found the secret.
Well, so in a way, I think if they see that you're passionate, that for sure it helps. You tell a story that kind of makes sense for them, and you can explain it in a way because sometimes it's too technical. Ours is quite technical, so you have to really break it down. That many of these investors coming from financial background, so to be able to explain to them that okay, what you're doing is why. So I would say that in our case, all our investors have some biotech expertise already, although not that's their original degree. So, but they are willing to understand, willing to take the effort to understand this. But still, you need to explain in the language that they will be able to understand and able to fall in love with it. And in principle, I think you also have to kind of make them kind of to fall in love with you and the team. So because many of the times, especially in early stages, the investors kind of more, they claim that, but I think it's kind of true that they invest in the team and partly in the project. And so the team has to be kind of convincing enough that, well, this is a team that I believe that will produce something. Even if it's not that what they promised, but it needs to be something where they will go.
I find this process particularly interesting. We talk about deep tech, biotech startups, and there is always the possibility to found something and go mainstream. And like artificial intelligence, currently everybody's doing something in artificial intelligence, LLMs, and you can do something, but that doesn't really go off. There's a lot of competition on the market, and the market is saturated in a bubble and up and beyond. With these deep tech founders, there is an interesting process. Um, they need to study some years. first become experts in their field, otherwise they're not taken serious. And then they need to identify a particular problem nobody sees. And this, I think, from the psychological point of view, is quite interesting because many humans would just say, okay, if nobody sees it, it's just a stupid idea. But the founders need to stick to that and
Yes.
say, I don't believe it's stupid. I think nobody sees it yet. And then they need to do the second thing, convince other people to join them when everybody else around them says, that's not gonna work. What's your secret? You have built a team, you have found investors. What's your persuasion secret?
Yeah, well, I think kind of showing passion could help for sure, uh, and showing credibility that you kind of you explain everything that what goes on, and then they could see hopefully transparently that what we're doing and where we're going and why you do what you do. So that's something which you need to tell to your people that they need to. You also kind of one of the top skills is to find the right people for the right job. So the job dedication is one of the things which kind of we reorganize a couple of times the company in this sense that okay, you better fit here, you better fit there. So that's for a while it's constantly moving, and then it's also you need kind of the people in that sense that they should understand that well you won't expect continuity here. It's every time is something new comes.
Yeah.
So that's something, but you have to show the confidence that okay, even if it looks crazy, there is somewhere we're going. So if we do not every time there is a new thing coming in, kind of in our case, what happened that we were developing a new version, and then there were a couple of pilot studies from pharma companies coming in, and then when a pharma company approached us, yeah, they would be interested to do this. Then kind of I had to stand out for everyone. Okay, drop what you're doing. Now we're dealing with this, and then that's kind of quick rearrangement because we had kind of contract with a pharma company where it was to show them that what we can do.
Mm, mm, mm. When we go back to your company, Cytocast, when did you make the decision? I mean, you had a failure behind you, it didn't really work out back in Hungary. When did you make the decision to do it again, to go into a
Yes.
startup again?
Yeah, so it's funny that I submitted two proposals to scientific funding agencies, kind of to do this project, and kind of it came back with the comments that yes, it sounds interesting, but we don't believe it's doable, and that's when we had this discussion that well, the governmental money is they won't willing to give into it because well, of course, there are some EU and other funding where they say they want these high gain, high risk type of projects, but this was probably even too much a much high risk project. And then we said that well, especially at that time in 2021, around that 2018, 19, and that time, there were more kind of startups popping up in these, and we've seen that maybe it will be easier to get funding from for such crazy idea from investors than from governmental funding, and that's so. Won't say we are forced into this, but we are forced to find some kind of solution which were outside of academia.
Wouldn't it? Why was it so difficult to find funding? I mean, isn't basically the venture case a crazy idea?
Yeah, so that's what happened. So finally, an accelerator was funding us. So that was I won't say relatively easy. I was talking to one of the angel investors who is kind of one of the top angel investors in Hungary in the biotech field, and then he was kind of introducing me to one of the accelerators, and then it was going relatively quick to get the first amount of money to start with, and then it was much harder to get to the next level, but eventually that happened as well. And now we are doing a bridge to go to the next level, and it's kind of yeah, in a way you are in a constant hunt for funders. I hope we in a year or so we will reach a stage then and we will have enough revenue that we can kind of smooth out for a longer time.
And then you can still look for new funds to scale faster and just
Yes.
accelerate.
Yeah, well, I'm dreaming of having enough revenue not to need that, but well, we'll see.
Yeah. In which year did you found Cytocast?
So it was founded. So it was founded twice. So it was founded 2019, kind of as a bootstrapping, just by the original founders. And in 2021, basically that's when we joined this accelerator, and that's when kind of we have accelerated the company, which is now called Cytocast Hungary. So that's the legal name, where kind of this company, which is owned by the founders, is one of the owners, and that's where kind of the new investors come next to this company. So the official founding date of the company is 2021, December, I think.
So you did your first steps before the pandemic, actually, into
Yes.
founding the company. How did you perceive the pandemic? Was it good for a company like yours, or was it problematic?
Yeah, well, so eventually, so in 2021, when we get the funding, then it's kind of what was the good part of the pandemic, that you could talk to investors online, and it's kind of speed up things. So that's kind of one of the advantages of the pandemic. Well, the lockdown was not really helping us, and actually at that point we were doing digital twins for a city. So we created a virtual town where we simulated that how the SARS-CoV spreads, and then what type of interventions can change this. So that was something which eventually it's a joint project of Cytocast and the university, where we created a digital twin of a town where we could test kind of interventions to stop the spreading of the virus. So that is somewhat related to this, but that's kind of off topic which came out of this.
Mm-hmm. And then I think it was November 30, 2022 was the genesis moment of large language models with ChatGPT going live for the broad public. What shift did you experience with your company?
Yeah, so the large language models, well, of course, one direction is really on the bioinformatics side, which is AlphaFold related, which eventually uses related technologies. So that is really a bioinformatics core question where we use these type of approaches. Of course, we use kind of really the ChatGPT and other approaches in assembling and collecting data from the internet in a way that is, and kind of, because for instance, one of the challenges that many of the clinical trials are just long verbal write down that how many people were involved and how many got placebo and such. So you have to digest all these, and that's where it helps a lot to digest this automatically and kind of create structured data from the unstructured clinical trials explanations.
And did you see any changes in the investment landscape with that? Is it now easier, harder for you to talk with investors?
Yeah, so I would say that there was a peak around 2022, 2023, so soon after, and then it was kind of the paradise for investment in this field. And but then the problem is that many of the companies where they invested at that point did not start to produce as well as they were wishing for. So now by 2024 and especially 2025 early, now there is much more skepticism. And now what they say that ideally we are over the valley and now it's kind of going up. We still need to see that if it's really constantly going up. But there was clearly a drop around 2024, early 2025 when investments dried out, and especially in this field, in this biotech field. So I was out in the US in January 2025. And I was showing this pitch of, well, what we're doing, we're looking for investment. and so on. And a bunch of investors said, well, 2-3 years ago with this one, you could easily get 10, 20 million. But some got 50 and they still don't produce enough data. So now we need, even for 5 million, we need you to show that you can produce revenue from this. So that's kind of the change in the investor mindset. And especially now there are uncertainties in the world in multiple aspects. So that's not helping either.
Okay.
So the wars here and there and political turmoil, that is making even more cautious the investors.
Yeah, currently, I think biotech, generally speaking, the investment is down to 2012 levels. And I think that your field currently tends more to resonate with biotech investors.
Yeah, so that's why we try to go more in the AI investors, because that is still going nice. Yeah, and especially what can be seen, that the money is still flowing in, but now it's much more the large deals. So successful startups get more money, more and more, but to start, so kind of C, D, maybe B level investments rounds are still going. A is less, C is even less. So that's where the problem is.
That's true. There was another point that I wanted to emphasize. You mentioned when we talk about forming a company, you mentioned passion and that the CEO has some basic understanding of finance and business. But when I look at the average startup in the biotech space, especially drug development space, I usually see teams that started in a lab, and you have a lot of scientists on the executive board. And when you look at the team, it's basically they all have the same education. Then I looked at your company in the preparation, and what I found very interesting, extremely interesting and fascinating, is that you already have a board with pharma business experts, and you consciously decided to do it that way. Why did you make this decision?
Because we lacked that expertise, and we really needed someone who can kind of streamline our ideas, and then to tell that, okay, well, this is nice what we produce, but how this will can be turned into real product that a pharma company might need. So that's why we have kind of, as I said, this earlier CEO of the second largest Hungarian pharma company, who kind of see from the above. And we also have probably the most successful medical chemist who was leading the development of a bunch of drugs at various pharma companies, but he's now back in academia. So kind of we have people, an advisor from both from the beginning and the end of the drug development pipeline. So, well, not at the end, kind of someone who sees the whole pipeline. So that's why we are really lucky to have really an all-star team in Hungarian drug system involved in our approaches.
I think this is one of the most important success factors that are completely underrated when you look in the startup world. Yesterday, there was this press release from Abivax. It's a French company. They had a gain in their stock price, I think, in a few minutes of 500 to 600% because of positive phase 3 data. And today I looked behind their facade and was thinking, why are they successful while others fail? And when I look at their team setup, they have exactly what you describe. They have strategy expertise, they have business expertise, they have market access expertise, they have clinical expertise, they have preclinical expertise. So basically, they cover the whole picture.
Yeah, we also have kind of the machine learning expert of this drug discovery field as a scientist, Alberto Pacanaro, and we also have an investor and the technology CTO type of advisor who is helping on that direction. So that's kind of the four wheels we are running on.
How do you identify the people that you need? What's
Yeah.
your process?
And actually, we have now a machine learning expert as well, who is more in the machine learning development. Well, these were all coming through some connections. So kind of some of these we approached ourselves. Some I knew before from my scientific side. Some were suggested to us by various people who looked at our company. So that was really helpful. And I, through one of our investors, who was kind of the lead investor and this earlier successful biotech person who sold his company to the Charles Rivers laboratory called Ernie Duda. So he helped me to get into the CEO CF program. I don't know if you heard of that. So that is a program where kind of C-level people are involved, and they have monthly online meetings and 3 times meetings in person during the year. Because kind of with the approach that the CEO is kind of the loneliest job, because you have to make all decisions on your own. And then what we have in those sessions, that someone comes up with a problem, and we have 1 hour that a board of 5, 10 C-level people are understanding the problem, asking questions, and then giving their suggestion how to solve that. And you learn a lot through all of this, also from your questions, your answers, and the others' questions and the answers and the situation. So even if it's not your problem. So this is kind of really useful, and I think I learned a lot from this, and this was also giving a bunch of connections. So this is kind of one of our board members coming from there, kind of now the salesperson who will now finalizing the deal to start our sales program is also coming from there. So that is really a useful source for me on the personal level, and also from the company level.
Yeah, I totally agree. Totally agree. Business is nothing that you can learn in theory. Business you can learn in praxis. It's like an art, and through conversations and talking with other people, business cases, this is exactly the way you should do it. When we stay with the pharma industry and talk a little bit about future business cases, we have now an artificial intelligence wave, and you operate in that space with your company. How do you envision the future of pharma?
Yes, so I think that there are a bunch of successes on the generative AI direction. So it will, like in silico medicine and others, where they can push their compounds to clinical trials much faster. So I can see that this is getting to the mainstream line. Well, I was discussing with Alex Zavorankov, the CEO of Silico Medicine. He was also saying that, well, he's publishing a lot on this field. He's making successful things, but still, kind of large pharma companies are still not treating him in that position as. So there are still some skepticism in larger pharma companies. So I believe that this is something which you can turn down. So actually, Alex, I know there is this Alliance for Artificial Intelligence in Healthcare. So one of these societies, if you wish, of companies who are interested in AI and healthcare. And that's where there were a lot of discussions that there are these companies who kind of, when we talk to investors, they are on the list of competitors. But actually, this could be much more of collaborators, because could be easier to convince pharma companies that AI approaches, and especially kind of putting all these companies in a pipeline, could really speed up the whole drug development process and the whole drug discovery process. And eventually, I think that's where we're going. How it will happen, if kind of larger pharma companies will start to buy these in, or they will just get these companies just get orders for larger pharma companies, or kind of they outgrow the large pharma companies. It's hard to predict, but I think this wave is coming. How it will splash, that's less clear, but somehow this will speed up the whole process, and especially speed up the process of finding, just as you mentioned, kind of new compounds working on new diseases. So that's where I think there is a large opening, where kind of it's not that what was happening for the last couple of decades, that there were a pool of compounds and then they tried all of them on a given disease and then they selected which might work the best. But now you really have the designed compound, and you can design this compound now for multiple things and much more detailed, and you can have a much wider selection of target. As we know more and more biology, we understand better that which could be the potential targets to look at as well.
That's a great perspective, and I think there is a lot of potential for optimization in the entire drug development process.
Indeed.
From the lab and up to the market. I think we still operate in a 5 to 20 plus years time frame with a lot of friction. And what I found interesting during the pandemic was that the pandemic with the mRNA vaccines showed what's possible on the regulatory side to push things really fast.
Yes, and now the regulatory side is starting to look more bright or exciting because FDA came up with a couple of releases recently that they will more and more try to replace animal tests with approaches one side on the organoid level and on the other side on the in silico level. So now there are some approaches where you can even have a pharma company dealing with a software company, and there is this model-informed drug development paired program where they kind of put together the software company and the pharma company to get through one in silico solution through the FDA approval that you replace, say, for instance, the metabolism of the compound, the pharmacokinetics, pharmacodynamics with a model, or some quasar results you get from a simulation or some AI approaches. In that sense, kind of the major question that how much AI and simulation. So I think the simulation is more accepted by FDA at this stage because there you know what happens. So that is there you see the molecular details and it's really a copy on the whole level of the real thing, as much as we know. And whereas with AI, it's so if you have an interpretable AI, that could help a lot. But that's where we had this discussion again with Alex that in the bioconvention that he said that for drug discovery and to identify that which is an efficient drug, you might not need to understand the mechanism. But I think for side effects, you might need to. Because if it works, in a way, you don't care how it works if it works. But if it works but causes something, then you might want to understand why it causes that. And can I avoid that? So that's where this might be a bit different.
Yeah, Alex is a great guy. He was on my podcast 2 years ago, and he's really doing amazing work. And when I think back to the 70s, I mean, also biotech, the development, biotech and drug development is undertaken is impressive. In the 70s, biotech didn't exist, and then there was Genentech, and Apple was new, Microsoft was new, personal computing was new, and nobody could imagine to develop drugs in computers back then. Even when I look in 2006, I think it was the year when NVIDIA started with their CUDA project that enabled a lot of future technologies and ideas, and probably also companies like yours. And now we are at the point where people like Alex and you actively think about how the future can look like. And I would like to hear your perspective when we go 20, 30 years into the future and think there are no restrictions. Everything's possible. Everything that needs to change, changed. How does the future in drug development look like, in your opinion?
Yes. Well, in fact, that just as you mentioned, like NVIDIA, now they even have a drug development part, so the drug discovery branch. So this is everyone is doing that, and that's why it could happen so fast, much faster than you expect to have kind of breakthroughs here. And I think that what will happen, so one thing for sure, that drugs will be personalized. So in a way, now there is a portion of people who have their genome sequenced. I would say in a couple of years, kind of everyone should have their genome sequenced. And if that's sequenced, and even if they have some other measurements on the person, then there will be enough data to tell that for this given person, what is the best treatment. So that will be a lot AI supported, that and various combination of various technologies, that if you walk into a hospital in, I don't know, 10, 20 years from now, you might not even need to walk in. You might do it. Yeah, in fact, you do it at home with your phone, which has a little sensor that you put it, I don't know, a drop of blood, or even at that stage, you might not need to make a drop, but it looks in under your skin. And then it makes some analysis, and your genome is already in the system, and then it makes an analysis and tells that, well, you have to take this pill, or which is most probably a combination of multiple compounds, especially developed for this given person, and this might be in the door with a drone in 2 minutes, and then you take it, and then you're cured. So that's kind of the futuristic vision, but I would say that that's the direction we're going to.
Yeah, it's fascinating. Fascinating. You see it coming, so it's not just theoretical.
Well, what is really limiting this to come is really the amount of data. So to make it more and more precise, but the amount of data that's pouring in from biology labs is really huge, and at this stage is more kind of limitation is kind of how to digest those amount of data. It's not the amount of data is the problem, is how to digest, but that is where ChatGPT and large language models could help to digest the data much faster, and that could make a major push to have enough data to do things which we cannot imagine at this moment.
What I can imagine, what might be interesting to hear for investors who listen to the live stream at this point is you have a clear picture of the future. You're working on current problems, that's fine. But in the next 5 to 10 years, how do you plan to position Cytocast in the market in this shifting landscape?
Yeah, so that's why I said that in a shorter term, we would go for these population level digital twins when we be able to predict clinical trials. But in the longer run, we could go to this personalized treatment suggestion system, and I think that's really the part which will scale and maybe combined even with a company who is creating the medical device which digest the data, create proteomics, transcriptomics data, and our tool might be running on the same machine, just splitting out that for this person, what is the best treatment. So that's really, again, a vision, but that's kind of the goal and the direction where, in principle, if our technology moves as we plan, and if there is enough data to train it into that direction, then this is something which I see reachable. And really, I think the challenging part is really to have enough data to train it properly. But as things speed up, I see that this might be happening.
Yeah, that's a great vision. It's a great vision. I hope it really happens in my lifetime. And
yes. Yeah, some people told me that, well, you have to live through the next 15 years. That's the major challenge.
15, not 50.
Yes, 15. So if you live through the next 15 years, then there will be cure for almost anything.
Yeah, so I'm 50, I have a chance to see it.
Yeah, I'm also 51, so yeah, that's our challenge, to keep in shape.
Yeah, that's true. That's true. It's very important. Attila, it's a great conversation. We're coming to the final minutes of our conversation.
Okay.
Before I'll ask my final 5 to 6, 7, 8 questions, is there a part that we didn't discuss yet that you would like to have addressed?
Yeah, I think it was so many things. I think we touched on almost everything which I would like to get through to this discussion.
That's great. For the final part, I would like to ask you some questions about future and your company. And the first part is you're currently solving problems and probably looking for partnerships and potential partners. What's your core focus? What kind of partnerships do you search for on the market?
Yes, so actually we have now formed an EU proposal group for to go into this direction, and actually to develop into this type of interaction to collect data. We provide a digital twin. So these type of partnerships, and that's what I mentioned, that either on the safety level, but also we might be interested to try that how our technology can be used to address other questions. So we had this idea to use this for diagnostics. So if there is some interest on these lines, that's one thing. The other one is that we still run a few pilot studies more on the drug safety level, because so if someone comes with a question which is not on the list, which I was mentioning, that how our tool can be used in drug discovery, we would be quite much interested if someone comes that, well, why don't you use it for whatever, and then we could solve other problems. So we are happy to discuss kind of new question that we might be able to address with our tools, or little modifications that enables us to that.
And when a CEO from a pharma company is listening, what's the reason why they should reach out to you at this point in time?
Yes, so basically to see how we can help them and to get their collection of compounds to us, and then we can do the screen and give a suggestion or ranking that which we might see as a better one or at least a good one to push through to the next steps in their pipeline. And basically to understand how our approaches work. So kind of really the goal was to get through the idea that how our technology works and how this could help them.
Mm-hmm. And you mentioned earlier from the investment side that you're also looking to raise funds. How is the round going? Is it open? Is it closed? Are there chances to get in?
So we are just closing a bridge round, and we are open, we're almost opening a new larger 5 million round, which would be kind of our seed round. We are now, that's important to tell, we are pre-revenue. So now we are negotiating with a couple of companies kind of that to have the first paying customer. So we had a bunch of pilot studies, which we still run, and then we'd like to turn these into paying customers. So we have now a very new version. So we will have the grand release in the beginning of September. When we go back to earlier customers, we look for new customers. And then kind of by the end of the year, we plan to get the first few paying customers. And as I said, we got back from a bunch of investors that, show us that there is someone willing to pay you what you do. So that's the idea, that once we have that, then we go back to these investors or anyone else interested to get kind of a larger round on the level of 5 million euros, which can help us to go into the population level development.
That's good to hear.
And expand on the sales pipeline. Yeah.
That's a good plan. And what's the best way to reach out to you? What's the best way to start a conversation with you?
Yeah, so if someone comes to our website, which is cytocast.com, or sends an email to info@cytocast.com, or reach me on LinkedIn, that's either of these can work, and we are looking forward to any interactions.
So LinkedIn, direct messages work also in your way, and you read regularly your messages?
Yes. So far I could handle.
That's good to
hear. Maybe
hear.
After this discussion, I will be overwhelmed.
Attila, thank you very much for your time. It was a fascinating conversation, and I really like the vision that you have for the pharma industry and drug development. I mean, it would be an ideal case 20 years down the road that you just go to the doctor and they produce exactly the drugs that you need in real time, everything simulated. It goes fast, no 5 to 10 years waiting, no 1 to 5 billion for a one drug cures all approach, but really personalized. It would be so great to see that and make it happen.
Yeah, that's the plan.
Yeah.
Thanks very much. Thanks for the great questions and the fascinating discussion. Thank you.
Thank you, Attila, and see you soon. Have a great day. Bye.
Thank you. Bye-bye.